Hi students, hope all are doing well. In the previous session, we discussed the cell division mitosis. Today we will explore another topic from the cell division, that is meiosis. So what is meiosis? What are the different phases of meiosis? We will compare the meiosis and mitosis. These are the topics we will cover in this today's session. Okay, let's start. So, meiosis is a special kind of cell division that occur in specific kind of cells. Okay, so let's start with the first definition of meiosis. Meiosis is a specialized cell division. In what type of cells it is there? It is taking place in the gametes. Okay, so the diploid cells in the germ cells undergo division to form haploid gametes because of this meiosis. And we know that the diploid cells, what do you mean by diploid cells? We know the full number of chromosomes. In the case of human, there are 46 chromosomes are there in the diploid cells. When it undergoes meiosis, which result in the formation of haploid gametes that consist of how many chromosomes? Half number of chromosomes, 23 chromosomes. So all of you learn what is that the definition of meiosis. It is a specialized type of cell division, occurs in the gametes. Okay, so the germ cells undergo division to form haploid gametes. So unlike mitosis we know in the previous session we discussed mitosis. They are maintained the same number of chromosomes. That's why mitosis is called a equational division, right? But here reducing the number of chromosome into half. So it is called a reduction division. Okay, that is one difference between mitosis and meiosis. So meiosis occur in the germ cells. Okay, so germ cells, these are the specialized reproductive cells. From that only gametes produced. And we know that there are two types of cells. We learned about the two types of cell. One is called a somatic cell. Somatic cell is there and also the germ cells there. Somatic cells undergo division and that division is basically mitosis division. Okay. But germ cells undergo division, that is meiosis. Remember these 2 points. Somatic cell division is the mitotic division. Germ cells undergo division, that is the meiosis. And this germ cells division result in the formation of gametes. What are the two types of gametes? The male gamete and the female gametes. The male gamete is called sperms and the female gamete is the ovum. So the sperms produced in the testis by the process of spermatogenesis is by meiosis. In females, the primary reproductive organ such as that ovaries, inside the ovary only, the primary oocyte undergo meiotic division to form the egg or the ovum, and that is through the meiosis. So where does meiosis occur? Remember, in the germ cells, exclusively in the germ cells, meiosis occur, which result in the formation of the male and female gametes. Clear? Moving on to next, what are the significance of meiosis? So meiosis is very, very important for maintaining the species. How it maintains the species? Because it maintains the same chromosome number. Each species, there is a specific chromosome number, right? What is the chromosome number of human beings? It is 46. So 46 chromosome number is there for the species Homo sapiens. For maintaining that species, there should be a specific chromosome number should be maintained. That maintenance is through the meiosis. Another significance is create the genetic variations. Genetic variations in the genetic constituent or in the genes, that causes the new characters and all. That is due to meiosis, specifically the crossing over and independent assortment that we will discuss in the coming part, right? Then it enables the sexual reproduction. So sexually reproducing organism for the formation of the haploid gametes, that is through meiosis. So these are the significance of meiosis. Then the key features of meiosis, the basic key features, right? So there are certain difference between mitosis and meiosis. So meiosis consists of two successive division, right? So two successive division means there is meiosis one is there, that is called a reduction division. In this, the homologous chromosomes separate. Then meiosis 2 is the second part of meiosis that consists of, that is the equational division. That is same as that of mitosis. What we learned in the previous class, the same way only meiosis 2 also happens. Okay, then there are four haploid products are formed. In mitosis, one cell undergo division to form two cells, two daughter cells are formed, right? But here in meiosis, what happens? There is one cell undergo first meiotic division to form two cells. It undergoes second meiotic division, right? So second meiotic division which result in the formation of how many cells? Four cells, and they are the haploid cells. Okay, so the mother cell is diploid and daughter cells are haploid in the first one. Then the same number of chromosome will be continue in the next daughter cells. Okay, so total four haploid daughter cells are formed from a single diploid parent cell. That is another feature of meiosis. And genetic recombination taking place in meiosis. Recombination means combining the genetic constituent of different chromosomes. That is taking place in the prophase 1. Okay, so these are the key features of meiosis. Mitosis, these features are all not there. Mitosis, it is the same genetic what is that, genetic components will be there, and two cells only there, only one division is there. Okay, but here two divisions there, meiosis 1 and meiosis 2. Then four cells are formed, and those four cells are having the half number of chromosomes, and in those cells the genetic content, some difference will be there. Okay, so in this slide, is there anybody is having any difficult word, or you could not understand any words like for example homologous chromosomes? Okay, homologous chromosome means we know that in our body, in our cells and all, the chromosomes are in pairs. We generally say there are 23 pairs of chromosomes. Okay, so the chromosomes are always staying in a pairs only. So first pair, second pair, third pair, like that. 23 pairs of chromosomes are there. Okay, so first chromosome one we are getting from father, this is a paternal chromosome, and the other one getting the from the mother, maternal chromosome, and these two chromosomes carry the same kind of genes. So the chromosomes carry the same structure, same kind of genes, and they have a same structure. Then we can call those chromosomes are the homologous chromosome. Okay, so homologous chromosome, I hope that word is word you understood. Now going to next, the detailed explanation of meiosis 1, meiosis 2. So meiosis 1 is a reduction division. In that there is a separation of the homologous chromosomes taking place, and the chromosome number reduced. Then the sister chromatids remains attached and result in the two haploid cells. These are the key events in the reduction division or the meiosis 1. In the meiosis 2, equational division is there. Here, the sister chromatids separate. Okay, so in meiosis 1, which one separate? The homologous chromosomes are separating. You know, now the homologous chromosomes are having the same structure and they are carrying the same kind of genes. Okay, suppose this is one set of homologous chromosome. then we can see another set of homologous chromosomes like this. So when you check the morphology of this chromosome, this is almost the same only. This is another set of homologous chromosomes. So when these chromosomes are in pair only, okay, and these pairs and all will separate during the meiosis one. Okay, that's what the first point. Here in meiosis 2, the first point is the sister chromatid separate. That means in the chromosomes we know there are two sister chromatids. See, this is one sister chromatid, this is another sister chromatid. These sister chromatids separate and form like a separate like this. That is happening in meiosis 2. Right? Then in meiosis 1, there is a reduction in the chromosome number. Suppose the chromosome number is 24 in a species, it reduces to 12. In humans, it will be 23. But in meiosis 2, whatever in the daughter cells there, the same number will be maintained. Okay, here the sister chromatids as such we can see there is no separation. But here in meiosis 2, the sister chromatids will be separated. Okay, so here two haploid cells are formed as a result of the second division. There will be total 4 haploid gametes are formed. Let's discuss more in detail meiosis 1. Meiosis 1, as we discussed, it is a reduction division. Why it is called a reduction division? Because as a result of meiosis 1, two daughter cells are formed having half number of chromosomes. That's why it is called a reduction division. Okay, so meiosis 1 or the reduction division again divided into how many phases? There are 4 phases there. Okay, what are the 4 phases? Prophase 1, then metaphase 1, anaphase 1 and telophase 1. Prophase 1. Here whatever that what we learned in the mitosis, almost the same features and all key events will be there. Along with that, the homologous chromosome pairing taking place. And there is a genetic exchange, the crossing over taking place in the prophase 1. In the metaphase 1, the paired chromosomes, they aligned in the metaphase plate. You know about the metaphase plate. Then in the anaphase phase, what happens? The homologous chromosomes, once they reach the metaphase, after that they separate, they move towards the poles. And then the last phase, telophase, they are the that nuclear membrane and the cell will like us, both the nucleus will go to both the poles. Okay, telophase. So that's what the overall stage meiosis 1, prophase 1, metaphase 1, anaphase and telophase 1. So let's discuss one by one. First, prophase 1. Prophase 1 is the longest phase in meiosis. This is one important question. Which is the longest phase in meiosis? That is the prophase 1. Okay, so it may takes for some 2-3 days or maybe week or many years also it will take. Okay, so prophase 1 is the longest phase and that consists of 5 distinct stages there. Okay, so this you have to learn in the correct order. What are the 5 different stages? The first stage is called a leptotene. Then second is the zygotene. Then third, pachytene. Fourth one, diplotene. And finally, diakinesis. Leptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, so let's discuss about leptotene stage. So leptotene stage, here the as we know that in the prophase generally the chromosome condensation begins, right? Here this is the very first stage of prophase 1 of meiosis 1. So chromosome condensation just started here. So before that the chromosomes are just like a thread like structure only. This chromatin thread undergo condensation. It is more thickened. The thickening of the chromosomes started in the leptotin stage. Okay, so DNA begins to coil, compact packaging of the DNA taking place, and chromosome slowly visible in under the microscope. In the chromatin stage, it is not that much visible, okay, but the chromosomes then it's slowly visible, and then the surrounded by the nuclear envelope there. That nuclear envelope will be as such. There is no disintegration of the nuclear envelope, and the centrioles are there that started moving towards the poles. The centrioles there that will move towards the poles just started. Okay, that's what the key events in leptotin stage. So remember, leptotin stage, DNA coiling and it begins to compact. Chromosomes become visible in the leptotin stage. Nuclear envelope will be there, okay, but the centriole started moving towards the poles. So that's what about the leptotin stage. Moving to the next stage, that is a zygotin stage. Zygotin stage, very important point there, the pairing of the homologous chromosomes taking place. Okay, so that pairing we are using a specific word, it is called a synapsis. So what is synapsis? It is the pairing of homologous chromosomes. It is taking place in which stage? It is taking place in the zygotin stage of prophase 1 of meiosis 1. And this pairing is highly specific. Only the homologous chromosome, the having the same morphology, same set of genes, so such kind of chromosomes only will align together and a pairing will taking place. Okay, so for that there are different stages there. Okay, very first they will recognize homologous chromosome, they recognize each other through the complementary DNA sequences. Then alignment of those homologous chromosomes properly they should arrange, okay, and then the pairing, and this pairing is due to a specific protein called the synaptonymal complex. Okay, this is important, this term is important. What is synaptonymal complex? This is a protein structure helping for the pairing of homologous chromosomes. So zygotin stage, we can see the synaptonymal complex due to that pairing taking. Okay, clear? Yes, then moving on to the next very important crucial stage of the prophase 1, that is packaging stage. Packaging stage. As I have told in the previous part, there is a crossing over taking place that causes a recombination. So recombination, recombining of the homologous chromosomes as a result of crossing over, that is in the packaging stage. Okay, this is another important question, especially in the previous competitive exams and all. They asked this question: the crossing over occurs specifically in which part, which stage? That is in the packaging stage. So in that only the genetic variation also generated. Okay, now let's discuss with the help of a diagram crossing, or how is the crossing over taking place. So you can see here, the first there is a homologous pair chromosomes there. Okay, suppose this one maybe we can call as the paternal chromosome, and this will be the maternal chromosome. Okay, and in the next stage we can see synapsis. What is synapsis? The pairing of the homologous chromosome, just they pair each other. After that, what happens in the next stage? They cross over each other, crossing over, okay, crossing over taking place, and there is a breakage of the chromosomal chromatids, and then they exchange their part. So the blue chromosome, they exchange the some part to the pink color chromosome. Okay, that's what the maternal and paternal chromosome, they cross over each other, and then they cut the part and then exchange their material. So now see the last chromosome, there is, and compare with the first one and the last one, you can see some changes in the chromosome structure. Okay, that is called the recombination. So now these chromatids are called recombinant chromatids. Okay, so this is important: crossing over, recombination, synapsis. These terms and all clear. I hope these terms and all clear. Okay, synapsis, crossing over, recombination. And what are recombinant chromatids? Okay, so once again, so first of all, during crossing over, there is a DNA breakage occur due to some enzymes. Okay, so the enzymes only causing the breakage of the DNA strand. After that, the broken DNA strand cross over and join with the corresponding strand from the homologous chromosome. So the chromosomes part, there is a breakage happens. Then after that, they will join with the next homologous chromosome. And another important thing, there is a non-sister chromatids only always, non-sister chromatids only always undergo crossing over. Okay, non-sister chromatids. We'll go back to the diagram again. So these two chromatids are called sister chromatids. Okay, sister chromatids. But when you see this one and these two chromatids we can call as the non-sister chromatids. Always remember the crossing over occurs between the non-sister chromatids, not the sister chromatid. These two will not cross over each other. There is no crossover. Only this one and this one cross over each other. Okay, that's what the non-sister chromatids. Remember that point also. So first a breakage, then they will exchange their material, and then finally the sealing of the broken part. Okay, so the DNA repair mechanism seal the breaks and creating a recombinant chromatids. So for this and all, there is a recombinase complex is there. Okay, different types of enzyme. The enzyme complex involved in this crossing over for the breakage, joining, and all. A group of enzymes involved. So this process typically occurs 1 to 3 times per chromosome pair, generating tremendous genetic diversity in the gametes. That is why sexual reproduction, as a result of sexual reproduction, the young ones are not the exact copy of their parents. We are not the exact copy of our parents because we have some new characters there, right? And that new characters is due to this crossing over and recombination. Okay, yes, so that's over. So which was that stage? The previous stage was the packetine stage. So how many stages we finished till now? Prophase, leptotene, zygotine, packetine. Next, the fourth stage is the diplotene stage. Diplotene stage, one of the key event is the chiasmata formation. So you may think, what is the term chiasmata? So chiasmata is nothing but there is a scar formed in the recombinant chromosome. Okay, so suppose the recombinant chromosome in this part only they exchanged with the another chromosome. There is a small scar will be there. Okay, this region. So in this region only the other homologous chromosomes will be attached. Okay, and that will be there in the diplotene stage that we can see clearly. That is, that region is called a chiasmata. Okay, so in diplotene, the synaptonymal complex dissolving. Okay, so what is the purpose of synaptonymal complex? That is for the pairing. Pairing done in which part? In which stage? Zygotine stage. Okay, after that, crossing over happened in the packetine stage. Then no more, they no need to stay together. So for that only the synaptonymal complex dissolved. Okay, so the homologous, after the dissolving, what happens? So definitely the homologous chromosomes start to separate. So when they are separating, there is a specific place where they already the crossover happened. Okay, that region they still remains there, okay, and that region is called a chiasmata. So the chiasmata is the visible evidence of crossing over events in a chromosome. If the chiasmata is there, then we can say that already crossover happened, okay, and it also hold the homologous chromosomes together, and each chiasmata represent at least one crossover event. Okay, so here the key event, just remember about the chiasmata. Then the last stage, but not the least of this prophase 1 is diakinesis. That is the final part of the prophase 1. The end of the prophase 1 is diakinesis. Okay. So in the diakinesis, already the major events over. What the changes in the chromosome to chromosomes, all those things happened. And now the surrounding part should be more some preparations required. Okay, so what is that? The surrounding nuclear envelope completely break down and then it disappeared. Then spindle formation also happens. Along with that, the chromosome condensation completed here. Okay, it become more thickened and easily visible. Then I have told you in the diakinesis, sorry, in the previous stage, what was that? The protein stage, that we can see the chiasmata, right? And here the chiasmata terminalization taking place. Okay, the terminalization of the chiasmata, this is important. Chiasmata move towards the end of the chromosome and then it go from the chromosomes. That is happening in the diakinesis stage. Clear? So chiasmata move towards the chromosome ends and maintaining the homolog connection. These are the key events in diakinesis. So prophase over, moving on to the next phase, that is metaphase. Metaphase 1. So in the metaphase 1, we can see the chromosomes are aligned in the equatorial plane. Like the previous one, you know that the same way only all the chromosomes are aligned. Okay, but here one difference is there when compared to the mitosis metaphase 1. What is that? Here in the equatorial plane, the chromosomes are aligned, but two homologous pairs aligned like this. This is first pair, then the second pair, like that. You can see the alignment. Okay, so this aligned chromosomes in the equator, that is called a metaphase plate. Okay, so metaphase plate is formed here in metaphase 1. The orientation of the bivalent. What is that bivalent? Bivalent means the two chromatids of the chromosomes called bivalent. It is random and independent. Each homologous pairs align independently. Like in the genetics part, we will study about the law of independent assortment. Okay, they align independently, the homologous chromosomes, and form the metaphase plate. So remember, metaphase 1, bivalents, not individual chromosome, they align at the equator. This is one of the difference from the mitotic metaphase. Mitotic metaphase, you can see all the chromosomes, they aligned in a single line, like this single line only in the case of mitosis. Okay, but in meiosis you can see there are two rows of chromosomes. How these two rows came? These two are the first homologous chromosome, then the second homologous chromosome, like that it is arranged. Okay, then in the anaphase 1, here the homologous chromosomes there. Suppose the homologous chromosome, this is one chromosome, this is another chromosome of the first homologous chromosome, then the next one. So anaphase stage, what happens? the spindle fibers will attach here and then they pull towards the pole. So this only the homologous chromosome separation taking place. There is no separation of the chromatids. Okay, paired chromosomes they separate. That's what here in anaphase stage. Okay, so homologous chromosomes separate, not the sister chromatids. Okay, only the chromosome the holes will go into the opposite poles. The sister chromatids stay together. But what happens in the whole cell, the chromosomes, suppose there are, now there are 4 chromosomes are there. Now, after the anaphase stage, what happens? 2 will be in one side, another 2 will be in the next side. So, half number. That's what the chromosome number is reduced to half. Okay, so here important point, there is no centromere division taking place. That's all about anaphase 1. Moving on to next telophase 1. So telophase 1, to haploid daughter cells formation, okay, the beginning. So in the cell, suppose in the cell you can see the chromosomes towards the poles, and then slowly whatever the prophase happened, the same opposite only in the telophase, slowly the nuclear membrane reappear. Okay, nuclear membrane reappear, and the chromosomes they become the chromatin thread. Like this, the chromatin thread formation. Threads will be formed, the chromatin. Then finally, it is going for the cytokinesis. What is cytokinesis? The cytoplasmic division. It result in the formation. So initially, the mother cell having 4 chromosomes there, right? From the 4 chromosome, now the daughter cells having how many chromosome? Half, only 2 chromosome. As a result of cytokinesis, cytoplasmic division, the 2 cells separated with the half number of chromosomes. So meiosis 1 is completed. Now the cell is ready for meiosis 2. But it won't go immediately to meiosis 2. There is a small pauses there, that resting stage is there. Okay, so this there is a small stage between the meiosis 1 and the meiosis 2, that is called the interkinesis. Okay, so interkinesis is different, the interphase is different. Interphase we learned in the previous class. And the cell cycle, what is interphase? Okay, here it is interkinesis. The period between meiosis 1 and the meiosis 2 is called interkinesis. So during interkinesis, chromosomes we can see partial decondensation is there. Nuclear envelope will be there. There is no DNA replication, no synthesis of DNA and all. Okay, and just preparation, metabolic reactions and all going on, and the cell is ready for the second meiotic division. That's all about interkinesis. And the last one is the meiosis 2. Meiosis 2, as we already know, that it is the equational division. It is same as that of mitosis. Somatic cells mitosis, same points only here also you are going to learn. So meiosis here also prophase to meiosis 2 only. So it is prophase 2, metaphase 2, anaphase 2 and telophase 2. Like that 4 different substages there. Here the chromosome number, whatever the daughter cell, now our daughter cell having the 2 chromosomes, right? From that 2 cells will be formed with the 2 same number of chromosomes. That's why it is called a equational division. So prophase 2, in that, so meiosis 2, prophase 2, there is a chromosome condensation taking place. Okay, chromosome condensation taking place, spindle formation taking place, and nuclear envelope started disappearing. Right? And in the metaphase stage is the second stage. Here we can see the chromosomes are aligned at the equator. So this is one homologous chromosomes. It's a homologous chromosome. Now it is in the another cell only. Okay, this is another cell. So here you can see only single chromosomes. In the meiosis 1, metaphase 1, there are two rows of chromosomes arranged. Okay, here only all the chromosomes are in a single line. Okay, so it is similar to the mitotic metaphase. So chromosomes line up singly at the cell equator. Spindle fibers attached to the kinetochore. We know the position of the kinetochore. It is next to the centromere. So all this spindle fibers that will be attached to the kinetochore. Then both the chromatids must be properly attached before the progression. Right? Then so the cell is ready for that. There is a tug of war is taking place. That means from this side, the spindle fibers will pull the chromatids. This side also they will pull. And finally, what happens? Finally, in the anaphase stage, this chromatids. Okay, so the chromosomes are like this only, and you can see the spindle fibers. They pull in the opposite direction. During the anaphase, what happens? The centromere splits, and then the sister chromatids they separate. Okay, so anaphase stage is characterized by the centromere division, a splitting of the centromere, which causes the release of the sister chromatids. But the number of chromosomes same only the sister chromatids separate. And then those sister chromatids move towards the poles, opposite poles. And equally, whatever the content will be distributed in the poles. Right? Then the last phase is the telophase. In the telophase stage, again, the decondensation of the chromosomes taking place, nuclear envelope reformation taking place, then the spindle fibers disappearance, and finally cytokinesis, the cell division taking place. Okay, so here in the mother cell you can see this is one cell. Okay, one cell undergo division. There is another daughter cells, and finally cytokinesis, cell cytoplasmic division taking place, and finally we will get two cells from here and here, another two cells. So total 4 cells are there, and 4 cells having half number of chromosomes. Okay, so meiosis, the whole meiosis result in the formation of 4 haploid cells from a single diploid cell. Okay, so that's all about meiosis different phases. Now let's for conclusion we will just compare the two types of cell division, mitosis and meiosis. We know that mitosis happening in the somatic cell, right? And meiosis happening in the germ cells. So the number of division in mitosis is only one time, but meiosis you can see meiosis 1 and meiosis 2. Okay. Then the daughter cells produced in mitosis, two identical cells. Only two identical cells are there. But in meiosis, there are 4 cells formed. They are non-identical. Understood why they become non-identical? Because of the crossing over. In which phase it is taking? It is taking place in the Pachytene stage of prophase 1. Okay. Then the chromosome number will be same only. A diploid cell, a diploid number of chromosomes there. But in meiosis, from the diploid, there is haploid number of cells, haploid number of chromosomes only there. And mitosis, there is no genetic variation. Whatever the chromosome, it become intact, and the same will be copied and given to the daughter cells. But here in meiosis, there will be high variations due to recombination. Here, crossing over does not occur, but in meiosis, crossing over occurs in prophase 1. And duration of mitosis is very less only as compared to meiosis. Meiosis is like a very long as compared to mitosis, and we know that which is the longest phase of meiosis. It is the prophase 1. I hope this session is very clear, right? So we will meet into the another session regarding the important questions related to this chapter. Okay, the cell cycle, mitosis, meiosis, and all previous year questions and all we will discuss in the next session. Okay, thank you all of you.